Hello, everyone, and welcome to today's Cryo-EM Current Practices webinar. You're joining the 3 NIH-funded National Cryo-Electron Microscopy Centers. My name is Sean Mulligan. I'm one of the scientists at the Pacific Northwest Cryo-EM Center. Also with us today are staff members from each of the centers, including my colleague here at PNCC, Marcelo DeFarias. From the National Center for Cryo-EM Access and Training, NCCAT, is Ed Eng, and from the Stanford SLAC Cryo-EM Center, S2C2, is Michael Schmidt. For our main event today, PNCC has invited today's speaker, Dr. Jennifer Cash, who is joining us from UC Davis. Before we give the floor to our speaker, we will have a quick update from each center on the resources we make available at no cost to the research community, and then Marcelo will properly introduce our guest. This webinar is a regular event we host the last Thursday of each month, highlighting users of the National Cryo-EM Centers who are kind enough to share the tips and tricks that have enabled them to make use of Cryo-EM in their research. You can save the date for our next talk on August 27th, titled Exploring the Cryo-EM Ecosystem, a career panel with former users and trainees of the National Cryo-EM Centers. Next month's talk will be different from our typical events and will feature 6 panelists from a range of academic and industry institutions. If you want to join us, you can already find the registration link on the events page at cryoemcenters.org, where you can always find registration for upcoming talks and recordings of previous talks. For those of you that will be at M&M next week in Milwaukee, Wisconsin, please feel welcome to visit the booth that will be staffed by members of the National Centers. Come say hi and find out more information. For final logistics today, if you have any questions about accessing National Center resources, please feel free to use the chat to reach out directly to any of the panelists. For questions for our speaker during the talk, please use the Q&A box to send your questions at any time during the talk, and we will hold them for a moderated Q&A at the end. Now I'll update everyone on what's going on at PNCC. PNCC has 2 types of proposals, screening access and general access. Screening access allows you to come on site for up to 5 days of sample preparation and screening with staff. This proposal is aimed at low resource institutions. For our other proposal type, general access, awards up to 480 hours of microscope time a year for 2 years. Approved general access proposals are also eligible for our training workshops, which cover topics from sample preparation through data processing. If you're interested in submitting a proposal, our deadline is the 1st of every month. We look forward to hearing from you. Next, I'll turn the mic over to Michael Schmidt from S2C2.
Thank you, Sean. Mike Schmidt from the Stanford SLAC Cryo-EM Center. We have a full suite of 4 electron microscopes and all the specimen preparation equipment. We engage in both service, that is data collection for people, screening for people to want to optimize their data, and especially training. We have special programs for training, including a weekly user meeting where people come to us with image processing questions that they want to have answered. And I would especially guide you towards our archive, which has some workshops that were created by and held at SLAC by the CryoSpark people. They have absolutely great information, and you can go over them time and again. And we have to go over data image processing questions. Join us at S2C2. Thanks.
Thank you, Michael. Next from NCCAT, Ed Eng.
Hi, I'm Ed, coming from NCCAT here in New York. I'm also joined by my colleague Eugene, who runs our grid preparation screening program. In addition to that, we offer high-end data collection and training. Sean alluded to a little bit that it is conference season. If you're going to be at M&M next week or just in Milwaukee, I would like to highlight that Claudia Lopez from PNCC is the guest speaker at the Women in Microscopy Breakfast Wednesday, August 5th. So if you're there, please do sign up. A couple other plugs. There is a lot of conferences coming up. If you're going to be looking for conferences in the fall, there's going to be an Integrative Structural Biology Keystone, September 28th at Colorado. The early registration deadline is today. And if you are interested in coming to New York, we also have another Integrative Structural Biology Symposium, October 8th. If you want to find out more, you can go to our website at nccat.nyspc.org.
All right, thank you, Ed. Let's get started. I'm going to stop my share and pass it over to Marcelo to introduce today's speaker.
All right, thanks, Sean. And hello, everyone, and welcome to our webinar series. Today, I'm happy to introduce our speaker, Jennifer Cash. She did her PhD training in biochemistry and X-ray crystallography at the University of Cincinnati. She then did her postdoc training at the University of Michigan, where she learned Cryo-EM and working with Mike Chow-Frocco. She's now an assistant professor in molecular and cellular biology at UC Davis, where she started her lab in 2020, and where she also serves as faculty director of their Cryo-EM facility. She enjoys working with trainees learning Cryo-EM, and she's one of the co-instructors for the Cold Spring Harbor Cryo-EM course that takes place each year. All right, and now she's going to share how high-resolution Cryo-EM is uncovering regulation mechanisms of dynamic macromolecular complexes. Jennifer, it's over to you.
Thank you, Marcelo. Go ahead and get started here. Okay, does everything look good?
Looks great. Yeah, I can see your pointer as well. That's awesome.
Fantastic. All right, thank you for that introduction. I am very excited to be here this morning presenting my lab's work on these single particles that I'm going to be telling you about. So kind of in general, my lab is really interested in how proteins are regulated through both autoregulation and by binding to other proteins and lipids of the cell membrane. So we are really interested in kind of understanding how signaling scaffolds assemble at the cell membrane and how that in turn governs cell migration. So these proteins that we work on also tend to be quite small and flexible, which also makes them very challenging in single particle cryo-EM. So specifically, we work on Rho guanine-nucleotide exchange factors, or RhoGEFs, which overall function to accelerate the exchange of GDP for GTP on these Rho family GTPases. So this has been the focus of my career for the last decade or so, and we're really, again, interested in understanding how they signal and how that signaling is regulated. So this signaling can be initiated through a variety of different factors, such as signaling by cytokines, growth factors, or GPCRs, which then through various mechanisms function to activate the RhoGEF, which can then activate small GTPases like RAC and Rho, which then go on to regulate a variety of different downstream effectors. Again, overall to basically regulate cell migration and a variety of other really important cellular functions. So it's interesting to note also that these RhoGEFs belong to this family called DBL-RhoGEF family. And there are about 70 different members of this family that in general signal through only about a dozen small GTPases. And if you look at the family tree, what you can see is that these family members tend to be highly diverse. So you can see that overall, they are typically larger proteins, and they have a variety of different domain structures within the different family members. So again, what we're interested in understanding is how these are regulated, basically. So the niche that we can have in our field is that we not only study these kind of smaller chunks of these proteins, but we also try to understand and study the full-length proteins in order to tackle these proteins in a very holistic manner. So my lab has for a long time worked on PREX subfamily proteins, but what I'm going to be focusing on today is a newer family member to us, which is a family member within the RH domain containing subfamily of RhoGEFs. Now, if we look at just a handful of these RhoGEF family members, what you can see from this domain lineup is that what they all have in common is this DBL homology domain, pleckstrin homology domain tandem. So this is what's referred to as the catalytic core of these proteins. And outside of that, these proteins are highly divergent. They have very diverse domain architectures, and they're typically rather large. So we're really interested again in seeing how these proteins are similar to and different from one another, both structurally and from a regulation perspective as well. And I am really excited today to focus just on this one family member called PDZ-RhoGEF. And this is actually the first time that I'm presenting this story on PDZ-RhoGEF. It's a project that is much newer in my lab. We're kind of wrapping it up, and it is not yet published. So PDZ-RhoGEF, or PRG, is a RhoGEF that, again, promotes cell migration. That's something that is, again, common within this DBO-RhoGEF family. And what we know about PDZ-RhoGEF is that it is necessary for cell migration through its function at the trailing edge of the cell. So PDZ-RhoGEF acts on RhoA, which functions at the trailing edge. So what you can see is that when you knock out RhoA in cells, and you activate cell migration through the ligand LPA, you get kind of this non-elongated phenotype. Again, because PDZ-RhoGEF functions at the rear of the cell to release the trailing edge and allow for cell migration. And again, this is something that is very common within the DBL family of RhoGEFs. So what we know about its expression is that it's widely expressed in the body, particularly in the nervous system. And we know that it's also heavily implicated in different types of cancers. So it tends to be overexpressed or mutated in different types of cancers, including breast cancer, lung cancer, melanomas. And this is also something that's very common within the DBL-RhoGEF family because of their effects on cell migration. So dysregulation of PRG is associated with increased invasiveness and migration. So it can lead to metastasis of cancers. And what's really interesting is that when you knock out PRG in mice, for example, there's no phenotype. This is, again, something common among DBL-RhoGEFs. Typically, when you knock one of them out, there's another RhoGEF that can kind of step in and cover the function of that RhoGEF. So because of this, it makes these RhoGEFs really attractive therapeutic targets because they can be targeted with minimal outside effects. So what we know about PRG from a domain perspective is that it's composed of a DH-PH tandem at the center. So again, this is the catalytic core. This is what binds to the GTPase RhoA. At the N terminus, there's a PDZ domain. This is followed by an RH domain. What we know about function is that it is most likely autoinhibited in the cytosol. The PDZ domain is a protein-protein interaction motif, and it's thought to bind to different GPCRs like LPAR1 and GRPR. The RH domain is known to bind to G-alpha proteins like G-alpha 13 and G-alpha 12 to activate the protein. And again, that DH-PH tandem is the catalytic core. So it's what's going to bind to and activate RhoA. So we're really interested, again, in understanding what are the molecular mechanisms behind PRG signaling and regulation. So if you take a close look at this domain schematic, which I have here shown to scale, it might look like kind of a disordered mess, right? So it has all these regions that are predicted to be unstructured. It kind of looks like beads on a string. That being the case, we are still really interested in looking at this from a structural perspective because there is some precedent within the family that it might actually be a well-structured RhoGEF. So this is an example of PREX, which my lab also works on. And this is a protein that I've worked on for several years. We have probably the most structural information on PREX as compared to any of the other DBL-RhoGEFs in this family. And when I first started working on this protein, it was actually thought to be about 50% disordered. So the entire C-terminus was predicted to be completely disordered. However, like I said, we now have a lot of structural information on this protein. What I have shown here is the cryo-EM structure of full-length PRX. And what we can see is that it's actually quite structured. And these domains within this protein really tightly associate with one another to form this well-defined structure. And in fact, only about 250 amino acids of the 1600 seem to be disordered or at least flexibly tethered to the rest of this protein. So like I said, there is some precedent for us believing that there might be more structure to PRG than what it looks like from the sequence perspective. Just as kind of a side note, most of the proteins, like I said, we work on are difficult targets in cryo-EM. So PRX subfamily members in particular have a very, very strong preferred orientation problem, which you can see here in the orientation distribution plot. That's also reflected in the FSCs from our cryo-EM structures. And if you look at the cryo-EM map colored by local resolution, you can see that these proteins are quite dynamic. So outside of the well-structured core, there are domains tethered to that core that are tethered quite flexibly, and that makes them very, very challenging cryo-EM targets. But again, what I'm going to focus on today is PRG, which is a project that was started in the lab by a very talented grad student, Matt Braga, who is a fantastic biochemist. So this is a project, like I said, that he started up, and he was able to figure out essentially how to make PRG in the lab. So we really pride ourselves on being very careful and diligent biochemists, and that is really, really important to cryo-EM in general. So we take both a divide and conquer approach and a holistic approach to addressing the questions that we have around RhoGEF regulation. So what I mean by that is that we study both isolated domains within the protein, but then we're also really good at making the full-length protein as well, even though they're quite challenging to express and also purify and work with, because they tend to be low expressors and also very labile. But this is a project, like I said, that Matt has pushed forward. He's also been working on this with a mentee in the lab, Emma Barpol. So the initial question that we chose to focus on was very specific. So we chose to focus on the function of this section of the protein called the GEF switch, which is 17 amino acids just upstream of that DH-PH tandem. And we wanted to ask a very straightforward question, which is, what is the contribution of the GEF switch to PRG autoregulation? So the reason why we chose to focus on this is because the GEF switch in other RH domain containing RhoGEFs is something that has been shown to be important for activity of the RhoGEF. So it's been shown in other RhoGEFs to somehow promote the activation of RhoA. But it had been very understudied in PRG. And again, we really wanted to know what are the molecular mechanisms that allow this really small region of the protein to have a really important effect on the activity of the protein itself. So in the beginning, to answer this question, we first looked at the structural information that we have available on PRG. So we have a few crystal structures of PRG, and these crystal structures are of the GEF switch or GS, including the DH-PH domain, and these are both bound to RhoA. So if we take a closer look at this structure, what we see is that the DH domain forms a alpha helical bundle. This is a very common structure in the family. And again, this is tethered to the PH domain. The GEF switch, again, is just upstream of the DH right here. And RhoA binds to the DH domain through its slightly concave surface. Now, there are two important regions on RhoA that I'd like to point out, and those are the switch regions. And these are the regions that engage the DH domain and they're really important for activation of RhoA. So like I said, we have a few structures of this complex of GS-DH-PH-RhoA, but we don't have any structural information on what this complex looks like in the absence of the GS, which makes it difficult to figure out from that structural information what the GS is actually doing toward activation. So what we can see is that the GS has really minimal engagement of RhoA. So what I have shown right here is that kind of close region between the GS and RhoA. And there are very minimal contacts. These are mostly van der Waals interactions, and there's very little buried surface area between those two regions. Instead, the GS primarily engages the rest of the DH domain. So it forms this hydrophobic pocket up against the first alpha helix, and then the alpha 2, alpha 3 loop of the DH domain. And that's where most of the packing occurs with the GS. So whatever it seems to be doing that through self-engagement. So again, we wanted to know what is happening to make this actually increase the activation of RhoA, since there are no direct contacts with RhoA itself. So again, we are focusing just on this shorter construct of PRG, and we wanted to take a biochemical approach to this, purify the protein, and then do a variety of different techniques to try to figure out what the GS is doing to enhance the activity of PRG. So Matt first went about purifying these constructs of DH-PH with and without the GEF switch. Sorry, my lights turn off. And what you can see is that he is quite good at purifying a very homogeneous sample of the RhoGEF, the DH-PH. You get this nice sharp peak on SEC. And he was also able to take those constructs and test them in our activity assay. So this activity assay, which I will show later on as well, is a MANT GTP assay, which essentially measures the incorporation of this fluorescently labeled GTP onto RhoA. And this is something that we can just measure essentially in a plate reader. So what he saw in measuring the activity against RhoA is that in the presence of the GEF switch, you get about 2 to 3-fold more activation compared to without. So that's a really significant increase in the activity. So we wanted to take this further and characterize the complex of DH-PH and GS-DH-PH with RhoA. So again, this is showing the purification where he gets nice peaks representing the purified DH-PH RhoA complex. So we first wanted to ask the question of, does the presence of the GS have any global effects on the structure of the complex? And we did this using SAXS, which are experiments that were performed at Sybils at LDNL. And what I have shown here are Guanine plots of the complex with and without GS. And what these essentially told us, based on the high degree of linearity and the residuals that are randomly kind of scattered around 0 in these plots, are that the samples are, first of all, of high quality. So the protein is good. What I have shown here are the Kratky plots from the SAXS. And what we see is that this bell-shaped curve here that peaks where these gray lines intersect essentially indicates that this is a globular system. So both complexes are globular, again, as we would expect from the crystal structure information that we have. And then looking at the scattering profiles and then the pair distance distribution, these essentially tell us that both complexes have a very similar global structure. So collectively, what the SAXS told us is that with or without the GS, the DH-PH RhoA complex is very, very similar from a global perspective. So we wanted to take this a step further and look more detailed at different regions of the complex. And to do that, we turn to a different solution experiment, hydrogen-deuterium exchange mass spectrometry, or HDX-MS. So if you're not familiar, this is a technique that allows us to look at exchange in solution. And we're looking at an exchange of deuterium or incorporation of deuterium into the backbone of our protein in solution. And to do this experiment, we essentially just take the protein, put it into a solution of deuterium, and then measure the exchange by mass spectrometry. Now we can do difference HDX-MS by taking two different complexes and then comparing them to each other. So this allows us to assess changes that occur when we have an additional portion in the structure like the GEF switch. It allows us to see what is different about the structure in the presence and the absence of the GEF switch. And these are experiments that we've done in collaboration with Sheng Li at UCSD, who's been a really long time collaborator of mine, and he's really excellent at handling our challenging protein samples. So this is one way of looking at the data that comes out of HDX-MS. And again, this is difference HDX-MS. So we're looking at changes that occur in the presence of the GEF switch. And what I have shown here are what are called Woods plots. And we're looking on a per residue basis, the differences that occur in the presence of the GEF switch. So the regions here that are shown in blue are residues that become protected when the GEF switch is present. And then red indicates that those regions become deprotected or more exposed or more dynamic when the GEF switch is there. So two regions that really pop out on this Woods plot are, first of all, the alpha-1 helix, which shows very, very strong protection. 50% protection is just a huge number in HDX-MS. And then also significant protection in the alpha-2, alpha-3 loop. So again, this is 10 to 15%, which is always also a very large change. So these are the regions that I showed you in the structure that are very close to the GS, and we expected these to be protected. Now, if we look at RhoA, the regions that show very strong protection are switch 1 and switch 2. So again, these are the regions that I told you were important for the DH domain to engage to allow the nucleotide exchange. And what was really surprising was the level of protection that we saw in switch 2. And this is simply because switch 2 is very distal from the GEF switch. So it's not close by, and the GS seems to be having some allosteric effect on the GEF switch or on the switch region that is very far away. So if I take that same information and instead plot it onto the crystal structure that we have, what you can see is this region here in the DH region, close to the GEF switch that is protected here in blue. And then if we look at RhoA, these are the two switch regions that I pointed out. So switch 1 and then switch 2, which again is very far away from the GEF switch. So this essentially told us that whatever effect the GEF switch is having on the activity or the activity of the DH domain is not just local, but it's actually allosteric, and it's transducing some effect across the structure of the complex. So these data are informative, they're good to know, but they aren't high enough resolution for us to be able to tell what the actual molecular mechanism is here. So what we next wanted to do was determine a structure of just the DH-PH RhoA complex without the GEF switch to figure out how it's different with and without the GEF switch. And we chose to do that doing cryo-EM. So it seemed like kind of a lofty goal to be able to image such a small complex in cryo-EM. So this is about 65 kilodaltons. Given the fact that we need high resolution information, to essentially see side chain detail. But we chose to pursue this anyway, just to see how it panned out. And these initial experiments that I'm going to show you are from data collected on our in-house Glacios. So this is with a K3. We do not have an energy filter. And here are all the details essentially for the data collection and the initial stages of processing. So to our surprise, we actually were very happy with how the DH-PH RhoA looked on grids. So this is not the best image. This is just an image that's filtered the way that CryoSpark typically filters them. But if you look at the denoised image, you can see that these complexes are actually really easy to pick out on the micrograph. And if you even zoom in, you can see the complex itself. You can see the individual components. So keep in mind that these individual components that you can see in this complex are each about 20 to 25 kilodaltons. So you can clearly see these complexes just on the micrograph itself. So again, we are quite happy about this. This is very exciting to us, and it made us very hopeful that we would be able to achieve high resolution with this data. So we, of course, collected a data set, took this through data processing, and these are the resulting 2D class averages. And again, we're very, very happy. These are fantastic 2D class averages. So you can see secondary structure details on these class averages. We could see exactly what we were looking at. So this kind of elongated structure here is the DH domain. And this structure right here is RhoA. So again, you can easily see the complex. And moreover, we could see that we had multiple views of this complex. So we didn't seem to have any orientation issues. We seemed to have a complete data set. So we took this through kind of a standard data processing pipeline and into 3D reconstruction. And again, we were quite happy with the reconstruction that we were able to achieve. So you can clearly see tubes of density for the alpha helices within the core. You can also see very clear density for some of the loops that engage the DH domain on RhoA. And if you kind of zoom in, you can actually see side chain density on the core. Here's an example where there's an arginine that's clearly out of place and should go right here in the density. So again, we were quite happy with this, and we wanted to try to push this to see how far we could get. However, what you probably noticed by looking at these images is that we are completely missing the pH domain. And not just weak density, we have absolutely no density for the pH domain. And again, remember that in the crystal structure, in the presence of the GEF switch, the pH domain is clearly present. So this is really odd to us. And we went back and looked at our 2D class averages. And we could see some classes where the pH domain is clearly there. So again, here we're looking at the DH and the RhoA, and this represents the pH domain. So this is exactly where it should be. It's the right shape. So this would be looking down the pH domain barrel. And we again see a couple of classes with this. However, it looks like the pH domain might be in different orientations in these different classes. Now, despite this, and having these 2Ds where we can clearly see the pH domain, we have never been able to reconstruct a map that actually shows any density for the pH domain. And that was really peculiar to us, which made us question things like, is the pH domain flexibly tethered? Which we didn't think it was, again, because we have crystal structures that all show the pH domain in the same orientation. And it made us question, is it actually denaturing? So it's possible that the pH domain is, in Cryo-EM interacting with the air-water interface and becoming unstructured, and we just never get resolution of it in our maps. So after seeing this, it made us want to investigate just the isolated pH domain further and characterize how stable is this domain. And one of the reasons why we wanted to look at this is because of all the previous experiments that showed the pH domain should be well-structured. It should be in a specific orientation relative to DH domain. So one thing that we did was look at the pH domain in differential scanning fluorimetry, or DSF. And if you're not familiar with this type of experiment, what we're essentially doing here is taking the protein, heating it up over time, and then essentially measuring the temperature at which the protein unfolds. And we can do this with a fluorescent tag. So when we do DSF on just the isolated pH domain, we can see that it melts at 47 degrees, which is a very high melting temperature, even for a well-structured pH domain, in my experience. Now, once we saw that it seems to be a well-structured domain, we next wanted to test the effects of detergents. So the reason why we wanted to do this is because we always tend to freeze cryo-EM grids with detergent. Our detergent of choice has been DDM. And the presence of detergent is the only difference between this protein sample and the protein samples that we've worked with previously, such as in SEC SAXS. So we wanted to see if that addition of detergent might actually be affecting the integrity of the pH domain. And what we saw from that was quite disappointing, because there are a few detergents that we've been using with these proteins that are quite detrimental to the protein itself. So again, we froze our samples in DDM, which you can see decreases the melting temperature of the sample by about maybe 5 degrees or so. There's also another detergent called CHAPS that we had actually been using in preparations of our full-length protein, which really severely affects the integrity of the pH domain. So that's good to know. We're definitely happy that we did this experiment because we no longer use these detergents with our samples, but it also allowed us to identify a few other detergents that seem to be not affecting the pH domain that we were able to test in cryo-EM. Again, because detergents we like to add because they give us nice thin ice. And thin ice is super important for really small particles in cryo-EM. So one experiment that we wanted to do after seeing this data is to just look at the DH/pH RhoA without any detergent in the sample. And this actually looks very, very nice. So again, if you look up close at this micrograph here, you can see the individual components of the complex. So the sample still looks really fantastic, even without detergent. However, what we noted is that Lubrizol still gives us thinner ice. And again, thin ice is really important for imaging these small particles. So we collected, again, most of our subsequent data with Lubrizol, but this data set that I'm showing you here is actually without detergent. So what we saw when we collected a data set without any detergent is that we were able to capture the pH domain. So what I have shown here is this would be DH/RhoA, and then this is the pH domain, which looks as we would expect for a pH domain. However, the additional caveat here is that in order to see the pH domain in its entirety, we also have to use continuous carbon film. So we think it's probably a combination of destabilization of the pH domain by detergent, but also just the pH domain itself inherently sticking to the air-water interface that's allowing us or not allowing us to actually see the pH domain in our data. So we haven't thoroughly tested the effects of the detergents to see if, for example, DDM on continuous carbon film would allow us to image the pH domain or not. We haven't fully answered that question at all. But what I can tell you is that the pH domain is definitely unstable to some degree in our cryo-EM experiments. And part of that is due to the detergents that we were using. So at that point, we had kind of hit a little bit of a wall with our data processing. Like I said, we need high resolution information to answer our question of what is the GEF switch doing at the molecular level to increase activity of the protein. And I showed you those tubes of density, and we were kind of stuck at that overall resolution. And the problem, bottom line, with imaging small particles in cryo-EM is essentially the low signal to noise ratio. And if you haven't seen it, Ollie Clark published a preprint last summer, I think it was, where he specifically looked at this problem in cryo-EM, and he came up with a data processing pipeline using high resolution heterogeneous ab initio reconstruction, or HR-HARE, where he essentially figured out ways where you can, in some sense, overcome this low signal to noise ratio and generate high resolution maps of small particles. And his method essentially was to do ab initio reconstruction using only data at high spatial frequencies. And he did this first in CryoSpark using a specific set of non-default parameters. And I'm not specifically going to go into those because, again, those were all published in his preprint. But using these parameters, we were able to overcome a lot of the problems that we were seeing in our low resolution data sets off our glacios. So this is the same set of 2D classification that I showed you earlier on. And again, initially, we were quite happy with this. We saw a lot of detail from these 2D classes. However, when we used the HR-HARE parameters in our 2D classification, we saw even better 2D classes with even higher amounts of detail. And importantly, those classes seem to be a lot cleaner. So typically when I see something that looks like this off of a first round of 2D classification, I will take that a step further and do maybe 2 or 3 additional rounds just to get a cleaner particle stack. With the HR-HARE parameters, this is a result that I would be very comfortable taking just directly into ab initio, depending on how clean the data set is. So again, we look at these particles, we see that they look fantastic. We have a really good orientation distribution. We can see multiple views of this sample. So I now just want to show you a comparison of kind of the standard data processing pipeline that we would use in CryoSpark on the left, compared to the processing that we've done with HR-HARE parameters on the right. And what you can see is we go from kind of this tubes of density to much more detail in our maps. So the maps definitely aren't perfect because at this point we had not refined our HR-HARE parameters that work specifically for our particle yet. But you can definitely see that the sample here on the left is much lower resolution compared to the detail that we can see in the map on the right. Another thing that we noticed here is that our alignments seem to get better. So what I have shown down here are the CFARs from the 2 different processing pipelines. And what you can see in the first one is a CFAR score that would hint at a preferred orientation problem. So with the CFAR score, typically the cutoff is 0.5. and above for having no preferred orientation problem. And if you're below that, there's some indication that you might have a preferred orientation issue. And we are definitely getting CFAR scores in our data processing that would suggest that, which is completely contradictory to what we are seeing in our 2D classification. So when those 2 things don't match up, you should definitely start to question where things are going wrong. When we started using the HR-HARE, our CFAR scores much more clearly matched what we were seeing in our 2D classification. So we went from 0.16 to 0.64. And again, showing that the alignments were actually getting much better in HR hair processing. I also want to point out here, I have the FSCs if you're interested in that. However, if you've noticed at this point, I haven't made any mention of resolution numbers. And that's because I think that resolution is a really poor metric of comparative quality between data sets like this. Because the resolution doesn't really give you an indication of whether or not your map interpretability is improving. But if you look back at our maps, again, they were much better, but we're not quite at the point where we can really see side chain detail. So again, we weren't at enough interpretability to be able to build side chains into this map right here with confidence. So we decided that we needed to take this a step further and actually collect data at a national center. And we decided to collect data on a Krios, although it's not because we thought we actually needed 300 keV data. We really wanted to collect data with an energy filter because we think that's probably going to make a big difference in terms of small particles. But we went ahead and collected data at PNCC on a Krios. So the data, again, looked really fantastic. We had a good biochemical sample. We see this nice homogeneous distribution of particles. And the 2D classification looked amazing. So you can see even better detail, as you would expect, from our samples on the Krios data. You can see really good detail on the side, or not side chain, you can see really good detail on the secondary structure. You can see multiple orientations of our particles. And again, we were very happy with this data set. Now we took this data set and we put it through the processing pipeline that we had figured out with our Glacios data. And then we continued to kind of fine tune the HR hair parameters. And again, I want to remind you that all of these things that we're looking at, essentially we're looking at just 45 kilodaltons of protein, and we're still getting these amazing images. So here's an example of taking that data set through that same HR hair data processing pathway, and then fine-tuning the Fourier radius step. So this is essentially fine-tuning the increment size between the spatial, or of the spatial frequency limits between iterations in the data processing. So if you take a look specifically at the pitch of the helices, what you can see is that the map on the right in gray has more detail in the pitch of the helices compared to, for example, this helix in purple. So Fourier radius steps seem to be an important parameter that we were able to successfully fine-tune to increase the interpretability of our map in this sample. So we took those set of parameters, did some more data processing, fine-tuning, and essentially this is the map that we've currently landed on of our 45 kilodalton complex, where you can see really good pitch in our helices. You can see side chain density. Everything fits quite nicely. Even the loops at the center of the complex are really well defined. And in the end, what seemed to be the most important for our data processing in terms of fine-tuning the HR hair parameters, where we're fine-tuning that Fourier radius step, fine-tuning the local refinement with the small search extent. So these maps have all been from ab initio straight into local refinement. And then masking also turned out to be really important for our small particles. So we really have to tightly mask these to get good resolution or good interpretability of our maps. Just to zoom in and kind of show you what I mean in terms of interpretability, here's an area right at the interface between RhoA and the DH domain where you can see the side chains in the data. So here's a lysine, for example. You can see good side chain density, also an arginine. So we're really quite happy with the outcome. from these parameters. So again, we're kind of at the stage where we can start model building, but we're not quite there in terms of the information that we need. So we're still doing a little bit more refinement of our data processing pathway. But what I can tell you is that we've done some further biochemical experiments to try to figure out what the GEF switch is doing. So looking back at the GEF switch and its packing against the DH domain, there are really a few core residues that allow for the GEF switch to interact with this surface. So those include isoleucine 805, glutamate 737, isoleucine 733, and this tryptophan 716, is really a highly conserved core component of this interaction. So we did a mutational analysis, and the essential takeaway from this is that both the interaction with the alpha-2, alpha-3 helix and the interaction with the alpha-1 helix are important for the GEF switch to enhance the activity of the DH domain. So again, we're trying to get this at a structural level to see what's happening allosterically, but at least locally, we can define this. And if we take these mutations, or if we take these variants that we've made and also look at them in DSF, we can see that their stability pretty well correlates with their effects on the activity. So we seem to be destabilizing the protein, which is in turn decreasing its activity. So here's our overall model for what we think is happening with the GEF switch and its effect on activity, where we think that in the presence of the GEF switch, it is somehow stabilizing the DH domain as a whole, which then stabilizes its interaction with RhoA to, in turn, increase its activity. So again, we're trying to figure that out at a structural level and fine-tune our structure to be able to pinpoint the interactions at the allosteric level, but we think this is what's happening. And I just briefly want to end by going through a little bit of the data that we've been able to collect on the full-length PRG sample. So again, we take a holistic approach to this as well. We're pretty good at purifying the full-length protein, and we're also interested in basically addressing the question of how do the accessory domains outside of that DH-PH core also contribute to its regulation. And one of the questions that you might be asking at this point is, can't you just use AlphaFold? Isn't that informative for how these domains might be interacting with one another? So just to show you the AlphaFold prediction of PRG, this is what it looks like. For the most part, it can fold the domains that we have structures for, but it is really poor at placing those domains relative to one another. And this is something that is really common across the DBL-RhoGEF family, where it tends to make structures that look very much like this. And what we've seen with PREX is that they tend to be very wrong in terms of how they place the domains relative to one another. So no, AlphaFold in this case isn't super useful. So what we've done is, again, produce the full-length protein, and we've started taking an integrative structural biology approach to address how all these domains might be interacting and regulating each other. So we're doing this using HDX-MS, cross-linking mass spec, and cryo-EM. And the experiment that we have done so far is HDX-MS, again with our collaborator Sheng Li at UCSD. So what I've done on this slide is to take that AlphaFold prediction and then color it based on regions that exchange very rapidly in the HDX-MS. So what I mean by this is that regions that exchange fully with deuterium within the first 10 seconds of the experiment. So those regions tend to be regions that are fully disordered in the structure itself, just because they're exchanging so rapidly. So that's what I have shown here. And those regions that rapidly exchange are shown in gray. And what you can see is that the alpha fold structure seems to be pretty accurate in terms of which regions are unstructured. And it actually seems to, maybe we didn't play, it actually seems to be over-predicting the secondary structure in different regions compared to what our HDX-MS data would support. So again, this is a sample that we're going to pursue in cryo-EM. I'm sure it's going to be super fun with all those disordered regions. But again, the end goal is to see if we can figure out how the structured regions are oriented relative to one another. Again, because that gives us information on regulation of the protein. But just to bring the story back to the pH domain, I wanted to zoom in on just the DH-pH tandem and the HDX-MS data that we collected on that in the full-length protein. And interestingly, what we saw is that the pH domain seems to have about half of the domain that exchanges fully within the first 10 seconds. Again, which might indicate that this pH domain is somewhat unstructured in the full-length protein. Again, this is something that we didn't expect based on the crystal structures that we have or the SAXS data that we have. And it's certainly something very interesting that we're going to look into further in the future. Okay, so main takeaways from this, assuming that you don't care about the biology of my system, which is totally fine. What I want to get across here is that in cryo-EM, biochemistry is definitely key. So I think one of the reasons why we've been able to collect such good cryo-EM data on this sample is because we are really good biochemists. We're very careful about what we do with our proteins and how we treat them. Another takeaway is to be mindful of the detergent additives that you use. So it's super common to add detergent to your samples before you freeze grids because it gives us that nice thin ice. But maybe if you're seeing a finicky protein, take a step back and try to assess what the detergent you're using might actually be doing to it. Here's something I might get in trouble for, but just a heads up, keeping in mind that I was a crystallographer in a past life. Crystal structures may not always be representative of the proteins in solution, and that is certainly something that we're seeing in our case here. Also, I encourage you to think about orthogonal approaches. So cryo-EM is really fun. It's fantastic to get the data you need, but then how can you support that with other structural biology approaches that you might be able to use the same sample on? And definitely 100% if you're working with small proteins in cryo-EM, take a look at Ollie's paper if you haven't already. It's a really great example of how we can push the limits of cryo-EM on small proteins. So with that, I will finish up and just acknowledge my group. Here we are at the Bay Area Cryo-EM meeting last fall. And again, most of this work was done by Matt and his mentee Emma in the lab, who've been really fantastic biochemists. And that's, again, the reason why we can push this project forward. I also want to acknowledge Faye, who is our director of our bio-EM facility. He oversees all of our equipment down there. And thank you for listening.
All right. Thank you so much, Jennifer, for this highly informative talk. We have a few questions here. I'll summarize too, because sometimes it's too much information. But could you please mention real quickly about what was the buffer used for your system and purification of cryo-EM, and also key points that you believe was the success for this project, like the detergent screening, as you mentioned, maybe grid types, parameters of 2D classification, like just to summarize and link questions to answers.
Mm-hmm. Yeah, so the buffer that we use is really standard. It's 20 millimolar heaps, pH 8. I think it's 100 to 200 millimolar sodium chloride, 2 millimolar DTT. That's the base buffer that we use. And then for the additives, just for the cryo-EM samples, we also use detergent, which the one that we've been using because it's stable for the pH domain is Lubrol. Initially, we were using DDM, which is the detergent that seems to be destabilizing the pH domain. In terms of the types of grids that we were using, initially we were just using standard quantifoil grids, just carbon grids. And that's what we got the first Glacios data set with. After that, we switched over to ultrafoils just because we were getting more protein in the holes with ultrafoils. And we also experimented, like I said, using continuous carbon coating on our quantifoil grids because that was what enabled us to recover the pH domain. But the data quality just wasn't as good with the carbon coating on the grids, I think just because of the extra background. So we didn't further pursue that. So the Krios data that we collected that I showed you the highest resolution structure from, the most interpretable map, that was with quantifoil and Lubral. And then 2D classification, I think was your other question. For our kind of pre-HR hair pipeline, we use just the standard small particle parameters in Cryospark. And then for the HR hair pipeline, we use the exact same parameters that are in Ollie Clark's paper.
Real quick, we're at an hour. I just want to make sure, Jennifer, that you have a few extra minutes to answer a couple questions.
Yeah, absolutely.
Okay, great. We'll keep recording the webinar, and any additional questions, if anyone has to leave, you can watch on the YouTube. But back over to Marcelo.
Yeah, thank you for the reminder, Sean. Another question here is did this protein require stabilization factors to be able to capture it stably, such as nanobodies, chaperone, proteins for expression, etc.?
Nope. We did not use any additives outside of what I told you was in the buffer. So no antibodies, nothing like that, no cross-linking. It's just a really good biochemical sample.
Great. Now I have like a question here is like brilliant work. The biochemistry microscopy were both highly advanced and impressive. How would you explain the tendency of PRG DH pH rho complexes to associate with one another and form rows or linear arrays?
So I don't know exactly that it forms linear arrays, but it does definitely associate with itself. So if you look at any of the crystal structures, and this kind of applies for the entire DBL RhoGEF family, if you look at the crystal structures of DH-pH or just pH alone, the pH domain always dimerizes it with itself in an anti-parallel fashion. So there is that surface on the pH domain, which is the exact surface that I just showed you here, actually. This gray surface is a dimerization interface. And again, it's an anti-parallel dimerization interface. It seems to be happening across the entire family. And we always see that in crystal structures that it's forming a crystal contact there as well. So I can tell you at least that in PREX, this is a really important surface for autoinhibition. So it's a binding surface for another domain within the protein itself. And it causes the protein to take on this kind of closed conformation. So I think that surface on the pH domain is probably just an underappreciated regulatory surface. It's sticky and it tends to cause this self-association.
All right. Still, Dimitri asks, in your workflow while you're still working on Glacios system, do you acquire again reference before starting a long data collection session?
Yes, every time.
All right, and he also says that I noticed density corresponding to only part of the H domain. How do you interpret this partial density, and how did you address it during data processing and in structural analysis?
Part of the DH domain. Let's see. I'm not sure which part we're talking about.
Yeah.
We definitely have pieces of RhoA that are missing, but I think those are kind of flexible loops. Most of the DH is accounted for in our maps. It's just the pH domain that seems to be entirely missing.
Yeah, sometimes questions are, written over the session. Maybe it was talking about when you're still showing 2D classifications where you're still missing a part, and could be this. All right, thank you so much, Dimitri. Another one. Okay, we did that. Do you think the pH domain might get away when the DH domain interacts with its partner like a switch? I wonder how the activity would be like if you didn't have the pH domain.
Yeah, that's a great question because I've been trying to convince Matt to do that experiment, but then we decided that it was too hard. The reason that we typically always include the pH domain is, well, a couple of reasons. Number one, the DH domain is a lot less stable without the pH domain, regardless of whether or not it's directly forming like a stable interaction with it. But because of stability, we always put the pH in there. And then the pH domain in some RhoGEFs also associates directly with the RhoA. And I think in some of the crystal structure data, the pH domain is forming some interactions with RhoA itself. Again, which is why we were expecting to see that in our cryo-EM data. So again, it's really surprising that we can't see the pH domain at all. Which makes me think that some of what we're seeing is maybe crystallization artifacts based on what I just said, like two questions ago. So, these are all things that we have to keep in mind when we're trying to, reproduce what we've seen in crystal structures, I think. Maybe what we're seeing is just from crystallization artifacts.
Great, cool. Couple of more. Excellent presentation, Professor Cash. I am a bit amazed why AlphaFold failed in giving most correct structure. Aren't there homologs available for this DH-PH Rho protein?
Yeah, so it does really well at predicting the DH-PH itself, just because it does have a lot of information on that. But really what AlphaFold fails on is predicting how all of the domains in the protein are situated relative to each other in the full-length protein, because it doesn't have information on how to place those. So like the RH domain, it predicts, we have lots of structures of that, it predicts that really well. PDZ domain, same thing. But again, because there's these really long loop regions in between, it just doesn't know how to form the tertiary structure, which again is important information when we're trying to figure out how these domains are actually regulating the DH-PH domain. So yeah, that's what I think.
Yeah, and just a follow-up question regarding the DH domain, how it interacts with its partner. As you mentioned, dimerization of the pH domain. Do you ever see a tetramer in your grids? Many thanks.
I don't think, as far as I can remember, that we've ever seen any sort of oligomer with PRG. We definitely see that with our PREX samples. With the pH domain, it dimerizes all the time. But maybe we just aren't looking for the dimer, and that's why we haven't seen it or any sort of oligomer with PRG.
Great.
All right.
Thank you, Jennifer, and thank you all for joining us today. We wish you all well in your cryo-EM endeavors. The National Centers are here to support your science, so let us know how we can help, and we hope you join us again. Thank you.
Thank you, everyone.